Okay. Students who are with Mr. Mohammed, I know that they have finished the chapter last week, but we have not finished the chapter yet. So as I'm teaching the second part of the chapter, we'll be hopefully finishing the chapter today, inshallah. So you all can just pay attention. This will be a kind of a revision for you all because this chapter will be included for midterm 3. Okay, so it will be a revision for you all, and that's it. Yeah. Okay. So, students who are with me, if you remember last week, we spoke about the different types of RNAs. We spoke about transfer RNA, we spoke about ribosomal RNA, we spoke about mRNA, we spoke about small nuclear RNA, okay? And today, the two RNAs which we did not speak about last week are your microRNA and the small interfering RNA. And I'm sure that you would have never heard the name before, small interfering RNA and the microRNA. Did you? Did you hear the name before? Never. I know, because yes, this is totally an unexplored area. Most of you would have heard the names of tRNA, ribosomal RNA, mRNA, small nuclear, microRNA, small interfering RNA. These three RNAs, they're kind of young RNAs, which were very recently discovered, right? And this is kind of, yeah, it's kind of an unexplored area. We're still exploring it, okay? Especially the microRNA and the small interfering RNA, okay? Let's try to pay attention. So when it comes to, I'm going to talk about both the RNAs together because both the RNAs share similar function almost. Okay. So the microRNA and the small interfering RNA together, these two RNAs are called as small RNAs. The reason why small RNAs, because they are not very long RNAs. Okay, they have shorter sequences, maybe like 20 to 30 base pairs. Okay, microRNA is just like 20 base pairs. Okay, and the small nuclear RNA is a little longer than it. It has 30 base pairs. So that's why the word together we call them as the small RNAs. Okay. Now, what is the function of these two RNAs? As I said that I'm going to talk about these two RNAs together because their function is almost similar. Okay. So their first and foremost function is they cause inhibition of the gene expression. What does that mean? If you remember, if you recall, we spoke about DNA forms messenger RNA, the central dogma. Messenger RNA is, once it's formed, it's sent to the cytoplasm where it is translated into protein. So all these diagrams that you see there, you're not going to prepare all these diagrams. They are just here so that I can help you in explaining what exactly it is all about. You don't need to prepare them for any midterm or a final exam, as you all are aware. Okay. So from DNA forming mRNA, mRNA forms the protein. This is the translation, right? So now what is the function of these two RNAs? These two RNAs, whether it's a microRNA or the small interference RNA, let's use the smaller terms like miRNA is the microRNA, siRNA is the small interference RNA. From now onwards, I'll call them as miRNA and the siRNA, okay? So whether it's the miRNA or the siRNA, both of them play a very important role in blocking the gene expression. What does that mean? That means these two RNA cause the silencing of the MR. As you can see here, there's a crossover here. What does that mean? That means if I have a microRNA or if I have a siRNA, these two RNAs have a capability of blocking the mRNA. If the mRNA is blocked, guys, can we get a protein from this mRNA? Will this mRNA be translated into a protein? Of course not, isn't it? So this is the way by which we are basically blocking a particular mRNA and that protein is not produced. Okay, this is basically a kind of a self-defense mechanism present in our eukaryotic cells, right, which simply prevent the formation or expression of any mRNA which we do not need. Okay, for example, now you might think that when will our cells use the microRNAs and the siRNAs? Suppose, for example, a cell is infected with the virus. Okay, let's say nowadays we all talk about coronavirus. Let's say a cell is infected with a coronavirus, right? You know that viruses, they do not have their own replicative machinery. They use our cells for the replication, right? And let's say if the cell is infected with the coronavirus, right? With the coronavirus, right? If it produces the mRNA, okay, so how is our cell going to respond? Our cell is going to produce the microRNA, it's going to produce the siRNA, and both these RNAs are going to combine with the mRNA of the coronavirus, and thus it will not allow the coronavirus to replicate in our cells. It blocks the expression of the viral mRNA. So both these organisms can target the mRNA. Okay. So in short, you can say you can say that they make a gene silent. They block the mRNA and prevent the translation of a particular protein. It's something like I have a in a cell, I have a gun. Okay. So gun hits a particular ball, for example, right? Or a particular target. So here in the cell, the guns which I have in the cell are either your microRNA or your siRNA. So these two guns are used to hit what? A target mRNA. What is a target mRNA? The mRNA which I don't want to get translated. For example, the coronavirus mRNA, for example, okay, or any other hepatitis A viral RNA, or any other Ebola virus RNA, any RNA, right? And let's say if this is the target RNA and I don't want this RNA to replicate in my body, so the siRNA and the microRNA are going to hit this mRNA, and it will not allow this mRNA to get translated. Automatically, no proteins are produced, no translation will take place. Is it okay? The basic mechanism of it, it's called as an RNA interference. Okay, we call this whole mechanism as the RNA interference. Okay, any questions till now? Did you understand the basic mechanism? Okay, now all you need to know is these are the only two paragraphs which you need to know, which you need to learn about the micro and the siRNAs. But trust me, if you read these paragraphs, you will not understand much from these paragraphs. So I got something called as a pathway or a diagram which can help you to understand the basic mechanism. But remember, you don't need to prepare this for any exam. This is just for the understanding purpose. You understand the concept and you can basically understand this whole page, okay, very clearly if you understand this mechanism. Are we ready? Everyone, are we ready? Okay, wonderful. Okay, let's pay attention. We are trying to know how microRNA or how siRNA are going to silence a particular gene, are going to prevent the translation of a particular mRNA, right? That is what we're trying to understand here. Okay, so let's say this is, okay, this mechanism can be, RNA interference can be present within the cell as a self-defense mechanism, as I explained you, if there's a viral attack in our cell, okay, or it can be, nowadays we are trying to make medicines, okay, based on this technology. Nowadays you will see that, you never know, tomorrow coronavirus, will be treated by the RNAi, treatment. You never know about that, right? There's people in the world somewhere, they are working towards all this. They've already found this mechanism effectful in the Ebola virus thing, right? In the influenza virus, some of the influenza viruses. So you never know, tomorrow using this technology, we are trying to make new medications based on the microRNAs and the siRNAs. Okay, so that's why as a anatomist, you have to know what is the basic mechanism, but I will not explain it in very detail. I will just explain you superficially, because if I need to explain this, I just need one whole class to explain what is RNAi mechanism. Okay, so I'll just explain you superficially so that you understand what exactly the mechanism is. Okay, so let's say I have a large double-stranded RNA molecule, okay, which is present in my cell that belongs to me, right? This is not the target mRNA which I want to kill, okay? This is how we produce the microRNA and how we produce the siRNA. Okay. So initially we might have a double-stranded RNA, especially in case of the siRNA. Okay. And there's something called as a dicer. So in simple language, you can say dicer is like a scissor. Okay. The way you cut your paper with the scissor, right? So dicer is like a scissor, it's an enzyme which cuts this double-stranded RNA into many fragments. So I have many fragments of double-stranded RNA. Okay, look at this. I have many fragments of it. And this is one fragment out of those. Okay, so the next step, what happens? One fragment out of it binds to a specific protein. As I said that we are not going into details. It's called a ribonucleoprotein. Okay, so once it binds with the protein, okay, it results in the formation of something called as a risk. Okay, if you go into your textbook, you can read more about risk, but that is not in your exam. So just to expand your knowledge, you can read more about what is the risk. It's there in your textbook. Okay, so what is a risk? Risk is RNA induced silencing complex. What does that mean? That means it is a protein. The brown thing which you see is a protein. Okay, and here which you see here, it is a single stranded microRNA or single stranded siRNA, which I basically got it from this double stranded RNA. Are we okay till now? We had a double-stranded RNA. We cut that RNA using a dicer into smaller pieces. Now we have a double-stranded small piece of RNA, okay, and it binds with a protein, which results in the formation of a RISC, okay, RNA-induced silencing complex, okay. What happens with this RISC, okay? This RISC is now ready. It's like, you have loaded your gun with the microRNA and the siRNA and you're ready to shoot. Okay. So what is your target now? Where are you going to shoot? What is your target mRNA? Anybody? What is your target mRNA? Where are you going to shoot? mRNA of the virus. Very good, Faisal Mohammed. mRNA of the virus, right? The mRNA, which is your target, which you don't want. Maybe that is a cancerous mRNA. Maybe that is a tumor-based mRNA. Maybe that is a viral mRNA, right? So that is our target. Okay, that viral mRNA is our target. So what happened to this? Okay, so this is my target mRNA, which I want to get rid of in my cell. This is my target mRNA, right? So what happens? This risk complex is now a shooter. It's ready with the microRNA or it's ready with the siRNA. So what happens? It binds to this target RNA. And the sequence here on this siRNA or this miRNA is exactly complementary to this one. Okay, so it's totally complementary to this one. And as the sequence is complementary, as you can see over here, what is happening? The maroon one is our microRNA or the siRNA. The green one is the viral RNA. So RNA, they know two strands of RNA, they join to each other. Okay. Now, in the cell, do we have mRNA as a double-stranded, guys or ladies? Is mRNA double-stranded? Never. Only DNA is always double-stranded. mRNA is always single-stranded, always, right? So as you can see here, this is a double-stranded mRNA and khalas, it cannot express now. Its translation is what? Stopped. Its translation is inhibited, right? It cannot express itself, okay? And what will happen now? The risk, the protein, the brown one protein, which was here, okay? This protein will cut this green one, okay, the target mRNA into smaller pieces, as you can see here. One fragment is this, another fragment is this, right? So what is the function of siRNA? The siRNA binds to the complementary sequence of the target mRNA, right? And it causes the destruction of the mRNA by forming smaller fragments. But like the way you cut a paper into smaller fragments. Similarly, siRNA will cut the viral mRNA into smaller fragments. And that's how the gene is being silenced. Now here comes the thin line of difference between siRNA and the microRNA. Okay, so microRNA does the same thing, but it does not cause fragments in the target DNA, target mRNA. As you can see here, do we have any fragments over here? No fragments over here, isn't it? No fragments at all. So in short, this is a very thin line of difference between the two. How microRNA works is that microRNA simply forms a kind of a loop over here. Do you see this loop? Okay, this is called a stem loop. So as a result of this loop, okay, the microRNA binds to the target RNA, and this also stops the translation of the mRNA. So microRNA does not cause fragments in the target mRNA. However, microRNA simply stops the translation of the target mRNA by simply complementarily binding to the mRNA and stopping the translation of that particular mRNA, silencing that particular mRNA. Okay, by causing an imperfect base pairing. This base pairing is not perfect. This is imperfect base pairing. Did you all understand the RNAi pathway, or did you find it interesting? If you found this interesting, then you're, you never know. Tomorrow, you as a doctor, maybe you'll do a research on, this is like, selling like hot cakes now, because all the medications, people are trying to, make the medications, the modern medications, the modern therapeutics, they are based on this mechanism. That's why, I had to explain it in detail, sounds very promising. As I said, that we never know. Tomorrow, using the microRNA or siRNA, they plan to make, inhibit the replication, or you can say the transcription of mRNA of the coronavirus. You never know. Right. Right now also there are many anti-cancer drugs. Okay, a lot of anti-cancer drugs are being developed based on this technology, a lot of them. Right, they basically are aiming towards silencing those genes, okay, which are oncogenes, which are cancer causing genes. Okay, there are a lot of work which is is being right now done based on this technology. Okay. And now you understood the difference between, right, this part of it, right? You understood the difference how uh, siRNA causes, the fragments. Okay, it blocks the translation, but what it does is basically leads to the destruction of this target DNA, right? But here, if you see here, there's no destruction of the target DNA, only gene silencing takes place. So this is basically what actually happens, but I don't know why your book is kind of, if you go to your diagram, and now everyone, I want everyone to pay attention. If you see in your book says something opposite, okay? But if this question is there for the midterm or the final exam, okay? Let's say the question says that the RNA which promotes degradation of the mRNA. What is degradation? Making the fragments, right? So you saw that, how mRNA was degraded. So we saw in this diagram, it is basically the siRNA which makes the fragments, and which is actually true. It's the siRNA which makes the fragments in the target mRNA. But your book says that it is the microRNA. Unfortunately, we cannot help with this. We cannot change the content of your book. So the question says like degradation of the mRNA is caused by, so please stick to your book. Even if it's wrong, to maintain the uniformity, we always stick to our textbook, even if the information is wrong over there. And if the question just simply says, which of the following RNAs inhibits the translation of mRNA? If you find both the options over there, microRNA and a small interfering RNA, you can choose both of them, right? Because both of them inhibit the translation of mRNA, right? But it's just that one causes the degradation and other causes the silencing of the gene. Okay. Are we okay till now? Did you all understand? If the question is there in the midterm, you know how to respond now, right? Yes, and this technology is also now currently being used, in the gene knockout. You know what are gene knockouts? Gene knockout means, for example, let's say, I don't know what is the function of DNA polymerase, let's say, okay? I don't know what's the function of DNA polymerase. So what do? I use this technology, I use the siRNA or the microRNA, okay, to inhibit the mRNA, okay, which forms DNA polymerase, okay, any particular enzyme, okay, and as I stop its production, okay, as I don't allow the mRNA to get translated, okay, I try to see that, okay, now there's no DNA polymerase. Let me see what effects will it produce in a particular microorganism. Okay, so those are called the gene knockouts. So this mechanism is also used by researchers to knock out a particular gene and to find out what effect it will produce if this gene is not expressed. Okay, is it clear, everyone? Okay, so structurally, if you talk about both the RNAs, structurally are similar. I told you the similarities between microRNA and the small interfering RNA, structurally they're similar. Okay, both have, this is 20 base pair long, this is 30 base pair long, right? Both of them control the gene expression, right? And both of them can form the risk, this risk, right? But the difference I told you, one basically causes the degradation of the target mRNA, other one simply causes the imperfect base pairing, and as a result the gene is being silenced. That was all about the RNA interference. Okay, now we'll talk about the exons and the introns. I think we spoke about it last time. What are exons, guys? What is the difference between an exon and an intron? I think when we were discussing the small nuclear RNA, I explained you the difference between the two.
Exon codes for protein or RNA, and introns, they don't code for anything.
Very good, right? So introns are which we don't need, right? Introns are the silent areas of a gene, right? The intervening areas which are not transcribed, which we don't need, the silent areas, right? And we want exons because exons are the ones which produce a particular protein or a particular gene. So we want exons, but we don't want introns, right? Because they do not function they're not translated basically. They're like silent areas. We cannot translate introns, okay? And we already discussed gene last time. I told you that gene is especially one fragment of a DNA. This is the DNA, right? If you remember last time also we spoke about it. Only that fragment of DNA, okay, which forms a particular mRNA or tRNA or a protein is basically called as a gene. Rest of the DNA is a junk DNA, the repetitive sequences like adenine, 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 thymine, 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 guanine, 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 right? The palindromic sequences. Okay. Now, I don't know why, for some reason, they have brought bacteria, here. But anyways, since it's there in the book, we need to know about it. So, bacteria and the eukaryotes. What is the difference between, genes in the bacteria and genes in the eukaryotes, like human beings? Okay. If you see, this is a bacterial gene, wherein if you see here, this is my gene A, which produces a protein A. This is my gene B, which not me, mine, the bacteria's gene B, which produces the protein B, right? This is my gene C, which produces the protein C, right? But do you find any introns here? Did you find any intervening sequences here? Of course not. Yes. So in short, you have to remember prokaryotic genes, they do not have introns. They do not have non-functional genes, right? And if you compare it with the eukaryotic cells, look at this. You compare it with the eukaryotic cells. Look at this. This is my exon. So this can code for a protein, right? This is a coding sequence. This is my exon 2. It can also code for a particular protein sequence. But then in between, what do I have? I have the non-coding area, which is the intron, which we do not require. So if you remember last time also we spoke about post-transcriptional modification. Remember the pre-mRNA when we were discussing the small nuclear RNA. Remember this diagram, guys? See, this is the pre-mRNA. Okay, the yellow ones are what? They are introns, and we don't need these introns, right? So when the pre-mRNA is produced, right? So after that, these introns are removed. If you remember, what is that process called? How do we remove the introns? Removal of introns from the pre-mRNA. What is the process called? Splicing. Yes, very good. If you remember, we spoke about last time, it's the splicing. Splice when we spoke about ribozymes, right? Splicing. Splicing simply means what? When you are removing the what? Introns from the pre-mRNA, right? So that you can have a mature mRNA without the introns. So it's just blue colored. It just has exons, okay? And then only this, mRNA, which is now ready to go to the cytoplasm, it is now sent for the translation. Okay, it's now sent for the translation. Is it clear? So if you see here, not only this, there are other things which also take place here. Okay, if you see here, there's something called as an RNA cap is added. We will discuss this in the next chapter. Okay, so before this initial or pre-mRNA is sent to the cytoplasm, okay, we add a cap to it on the 5' end. And we also add a long adenine tail to it at the 3' end. Okay, so this capping and this tailing is very, very important, because this capping and tailing will basically protect this mRNA from degradation, because mRNAs, they get degraded very quickly, right? If you are working in a lab, and if I give you, okay, this is the blood, this is blood, isolate the mRNA from this blood, okay, and then I give you another sample of blood and tell you, okay, isolate the DNA from this blood. If you leave mRNA in one test tube and leave DNA in one test tube and go to home, and next day when you come, you see that most of the mRNA which I have kept in the tube is degraded. Okay, while as that's not the case with the DNA, right? So mRNA is very, very sensitive. So that is why we need to add the poly A tailing here, so that we can prevent it from being degraded when it reaches to the cytoplasm. Okay, and the capping we add here so that it can facilitate its transport, okay, to the ribosome. But this is something which we are going to learn in the next chapter, not in this chapter. Okay, I just thought of adding it here. It's okay, Yomna, no issues there. Okay, so this is the diagram in your book. Look at this. So on 3' end we have the poly A tailing, and here we have the capping. And look at this, the final mRNA has no introns. Okay, it has just the exons, and it will be sent to the cytoplasm for the translation. Any questions till now, guys? Are we okay till now? Everybody all clear? No questions? Okay, if you have, please stop me in between. Okay, just let me know if you have questions. Okay, moving on further. Okay, we'll talk about something called as a satellites. Okay, so even though this topic is actually kind of very vast, but we'll just be confined to your book. We'll just try to know whatever is there in the book. Okay, so if you remember, I told you just now that most of the DNA in our body is just a palindromic sequence. What does that mean? That means just a waste DNA, repetitive DNA. Okay, as I said, it is adenine, thymine, guanine, right? So only 3% of the DNA codes for the proteins or codes for any of the mRNAs, mRNA, tRNA, ribosomal RNA, right? Small nuclear RNA, right? SNRPs, which we call as, right? Or microRNA. Now, we already spoke about introns are non-coding sequences, right? And then we have satellites. So what are satellites? Okay, satellites are again non-coding sequences in our body. Okay, you will usually find them on the chromosome. Okay, let's say if I have a chromosome like this, okay, okay, you will find these satellites or large satellites or repetitive sequences. For example, you will have, let me use another color here, you can have it, for example, at this end I have adenine, 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 adenine. Okay, maybe at somewhere in the middle I have guanine, 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 guanine. Okay, maybe here also I have uh, cytosine, 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 right? So on a chromosome, you will find these non-coding sequences, okay? Or you will find some of them here even in the centromeres as well, like guanine, 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 right? So why do we have these coding sequences? As I said that I will not go into details of it. I'll just tell you superficially what it is, okay? So for example, you have something called, this is called the telomere, the red one which you see here, okay? This one is called as a telomere. Okay, and in the telomere, which is at the end of the chromosome, you'll find a lot of satellite areas, right? Lot of repetitive sequences like adenine, 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 thymine, 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 right? And as they're present at the end of the chromosome, their main function is to protect the ends of the eukaryotic chromosomes against degradation. Okay, like for example, when you wear your shoes, okay, what your laces have plastic tips, isn't it? You might what are laces? The shoe laces, right? They have plastic tips at the end, right? And those plastic tips at the end, they kind of protect the laces, okay? Otherwise the thread will come out, okay? Similarly, we have telomeres at the end of the chromosome, which are nothing but the satellite areas, okay? And if you see here, look at this, they're repeated, plenty of times they're repeated, okay? And their main function is, they're just non-coding areas. Their main function is gene protection and stability in the chromosome. That is it. Whether it's a satellite, whether it's a large satellite, or whether it's a mini satellite, okay? For example, mini satellite is like, it's very small, like 10 to 100 base pairs long. That is it. Okay, so it's so very smaller, and the larger ones can be a little longer than that. Okay, and even we also have a micro satellites, which are like 1 to 10 base pairs long. So in short, you have to remember that when they're present in the chromosome, okay, their main function is to stabilize, okay, or to protect the particular gene or a chromosome. Okay, so even in the centromeres also, you'll sometimes find these repetitive sequences, and that centromere is the region where microtubules are attached, right? So simply remember, satellites are the non-coding sequences whose main function is to protect and to stabilize the chromatin or the chromosome. That is it. Okay, and the micro ones, they have been found that if they mutate, even though they're non-coding sequences, okay, we found them as a junk DNA, but we have seen that if they mutate, if there's a tafra there, okay, it can cause the cancer. Okay, so that shows that even though they're repetitive sequences, but they might have some kind of function. Who will answer this question? Explain why most eukaryotic genes are longer. I just need to say, just stop it over here. Are longer than the mature mRNA, right? The question should be like this, then the mature mRNA. Who will answer this question? Because of the presence of introns. Absolutely, very good. Because in prokaryotes, we don't have introns. Super, very good. Name the types of RNA involved in protein synthesis on the ribosome. Who will answer this? Very easy question. mRNA, okay. Then tRNA, okay. Then ribosomal RNA. These are the three main things. Then, of course, we know that in order to convert the mRNA, pre-mRNA into final mRNA, the mature mRNA, we also need the ribozymes. We need SNRPs. We need small nuclear RNA, right? But it's saying only up to the ribosomes. Look at the question carefully, okay? It's so then comes the next topic called as DNA replication. Okay, so before we start this topic, okay, I would like all of you to see a video, and then I'll start explaining it, because seeing these things, animated will definitely make a lot of difference. Okay, give me a minute. I'm going to upload a video here, and then I'll start explaining. So give me a minute. Let me know if you're not able to hear the sound, or if you're not able to watch the video. Where did the video go? Okay. Give me a minute. What happened? Guys, I'm trying to upload the video. Tell me if you're not able to hear the sound.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands.
Excuse me, doctor.
This unzipping is
We
done
can't
by
see the video.
Sorry?
We can't see the video, only the audio.
Oh, I'm so sorry. I thought you were able to see the video. I'm so sorry. I can hear but can't see. Okay. Okay, give me a minute. What happened? Okay, give me a minute. Mhm. Mhm. Let me try to play another video.
When DNA
Guys, I'm trying to upload the video for some reason. Give me a minute. I cannot. What's the reason? Mhm. Okay. I'm going to try for one last time. If it works, fine. Okay.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A.
Okay, ladies, unfortunately, I'm not able to play the video. I don't know what's wrong with it. But I'm going to do because I had basically planned to play a couple of videos, like 3-4 videos. Okay, what I'm going to do is basically I'm going to upload them on Moodle. Would that be okay for everyone? Would that be okay? I'm sorry about that. I don't know what's the reason. I cannot find the sharing window when I'm trying to share these. Okay, so I'm going to upload it on the Moodle today itself. Okay. So let's start with the DNA replication. Okay. So how many of you know about the DNA replication? What is replication all about? Yes, Zoom is totally unpredictable. Yes, Adil, I will. Don't worry. Okay. Yes. Can anyone define what is DNA replication? And what is happening in DNA replication? Copying of the DNA. Very good. Why do we need to copy DNA? Why do we need to copy DNA? Replace the DNA, producing 2 strands of DNA for mitosis. Very good. To make proteins, no. For cell division, Very good. For cell division, for growth, very good. Absolutely. Because you know that, see, DNA has to replicate. Replication of DNA is very, very important. Yes, yes, new cells, right? Not to make the RNA. Replication does not make the RNA island. Replication simply means that the separation of two strands of DNA will take place, right? And two identical DNA molecules will be produced. To make the mRNA is transcription, island, right? Not island, Lara, okay, right? So DNA is simply copying its own DNA. That is it, copying its own strand, okay? And why do we need to do that? Because we know that when the cell divides, okay, the cell has to divide, okay, if this cell has one molecule of DNA, and from this one cell at the end of the mitosis, we get two cells, right? And what are we going to give to these two cells? We need to send, we need to supply, or we need to transmit this DNA to the next forthcoming generations of the cells, right? So what do we do? So DNA replication will take place in which part of the cell cycle, guys and ladies? Chapter number 3, last semester, DNA replication takes place in which phase of the cell cycle? Does anyone remember from last semester? No, S phase. Yes. In the interphase, in the middle of the interphase, it takes place in the S phase of the cell cycle. S phase belongs to the interphase, not the mitotic phase. Because by the time cell reaches to the mitosis, the DNA has already duplicated. That is why, if you remember last semester, we spoke about that is why we have a duplicated chromosome in the mitosis, right? But before S phase, the chromosome might look like this, which has a single chromatid, right? So duplication of the chromosome will take place in the S phase. That means the replication will take place in the S phase. So in order to send DNA to the next generation, what do we need? We need to replicate the DNAs, right? So these two DNAs will be separated, and each strand will copy its own, its own complementary strand, right? As a result of that, at the end, we will get what? Two molecules of DNA, which we can happily send to this cell and to the other cell, right? So that is why DNA replication is very, very important. And what happens at the DNA replication? In the DNA replication, the two strands are separated to form the two identical, right, DNA molecules. So these molecules look like they're what? Are identical to their parent cell. Okay. That is about the introduction to the DNA replication. We had, we had two important people like Michelson and Stahl, right? In 1958, they proved that DNA replication takes place in a semi-conservative way. Okay. Does anyone know what is a semi-conservative way? What happens in the semi-conservative way? In the semi-conservative way, if this is the parent DNA, yes, let me see who has the answer. No, we did that in last semester, I think. Semi-conservative means that if this is my parent DNA, let's say this is the parent DNA, this is present in the parent cell, okay, what's going to happen here? Okay, in the DNA replication, only one strand is original. No, it doesn't mean identical. One new strand and one old strand makes up both the molecules. Absolutely, guys. Yes, you're right. So semi-conservative means that if this is your parent DNA, look at the color coding over here. These two strands will be separated. So this will be my one parent strand, and this will be my another parent strand. Okay, one strand will form this strand will form its own new what? Opposite strand, right? The complementary strand. Okay, and similarly, this strand will also form its what? new strand. So what is semi-conservative? Semi-conservative means one turn of the template. No, semi-conservative means that in the daughter cell, you will always have one old strand which is preserved. Okay, every time a DNA replicates, you will have an old strand which basically came from the parent. You will have an old strand which basically came from the parent, and then these old strands, they formed their own new strands. So this is my new strand. This is called as a semi-conservative model of DNA replication. This was proved by using radioactive isotopes. This was proved by Michelson and Stahl in 1958. So semi-conservative means every time a DNA replicates, you will always have one old strand and a new strand, one old strand and a new strand, one parent strand and one new strand, one parent strand and new strand. This is called as a semi-conservative model of replication. Are we okay? Everyone, is it clear till now? What is semi-conservative model of replication? Clear. Okay, because there were other models at that time. Some of them said that when this happens, okay, I don't want to go into details. Some of them said that no, the old ones come together, okay, and then the new ones come together. But it was Michelson and Stahl which said that no, DNA does not replicate this way. DNA replicates by this way. Okay. So remember, every part of DNA replication is a base for the next concept. Okay. So please pay attention, everyone. Okay. So we discussed last time that DNA in the chromosome has two functions. Our DNA has two functions. One, it causes replication, right? It produces the same copy like itself. What happened to this now? Okay. And for some reason, my Zoom is not behaving normally. I was trying to write something, but nothing was coming up. Okay, anyways. So DNA can cause replication. Okay, at the same time, why do we need DNA? We also need DNA to form new mRNA, to form the mRNA, so that mRNA can form the proteins, right? This is the reason why we have DNA present in our cells, basically for the replication and for the transcription to take place, right? And so that we can send this DNA to the next generation, and that will happen only because of the replication. Now, there are certain rules for the replication. Now, I want everyone to pay attention. This is so very important, okay? And I have seen every time, whenever we give questions, people mess up with the replication, you know? And this is such an important topic wherein we might ask plenty of questions, and I've seen students messing up with the replication, okay? So please pay attention, everyone, okay? Replication begins at a particular point. When the DNA is ready for the replication, okay, in the S phase, okay, replication cannot start anywhere on the DNA. It starts at a particular point, and that point is called as a origin of replication, or we simply call it an ori site sometimes. And this region is usually very rich in adenine and thymine. Because if you remember, there are only two bonds between an adenine and thymine, and there are three bonds between guanine and cytosine. So usually at the ori site, you will see that it's very rich in adenine and thymine. Are we okay till now? Okay. So it will be easy to break two bonds rather than breaking the three bonds. Okay. Now, once the DNA starts at the, so like, for example, if you see here, this was the DNA, right? So this is the point of origin. From here, the replication will start, okay? So replication can be unidirectional. It can be bidirectional. It's basically bidirectional, you know, in eukaryotic chromosomes. Like, as you can see here, it can move on this side. It can also move on the other side, right? I'm sorry, you know, if you see here, let me erase this. If you see here it's unidirectional because the fork is moving only on this side, okay? But if you see here, the replication is moving on this side as well as on this side. So it's usually bidirectional in the eukaryotic chromosome. Now you might be wondering, what is a replication fork? Okay, so these kind of definitions you have to know. What is a replication fork? Okay, replication fork, as you can see here, is this is a fork. I have a better diagram for this. Look at this. This is a fork which is formed where the DNA is opening up. Okay, and this fork is the place where right now replication is taking place. Are we okay? What is a replication fork? So as the DNA replication starts, okay, DNA strand is usually, it is in helical manner, right? So we need to unwind the DNA, and as it's unwinded, right, when the replication begins, okay, it results in the formation of a replication fork. So the question says that what is a replication fork? You will say that it's a point where replication is actually taking place at that particular time. Okay, so that is the, or you can say that the two strands of DNA, DNA helix, they separate at the replication fork, as you can see over here. Is it clear about what is a replication fork and what is the origin site? Any questions about it till now? Okay, and we said that replication is bidirectional. As I showed you here, it can take place with the same speed, but in the opposite directions. Okay, and it takes place, simultaneously. Okay. Did I explain, Mr. Akazi, about the discontinuous yet? We are going step by step. Okay, we don't want to mess up everything. We'll go step by, because the whole process is so very complicated that, if I end up explaining everything, you might be confused. So we are going step by step. I didn't even explain about the discontinuous yet, Mr. Kazi. Miss, where is the origin? Origin can be any point, Faiza. There's no predefined area in the origin. Origin is simply a point where the replication starts. That is it. Okay. And as I told you that usually at the origin, they are rich in adenine and thymine, because it's easy to break adenine-thymine bond than the guanine and cytosine bonds, which are 3 bonds, where we have 3 bonds together. Okay. Okay. Now we'll move on to the next step. What is the next step? Next step is that the two DNA, we will describe the process after some time. We are just trying to learn few basic points about the DNA replication, and then we will move on to the enzymes part and how it's unwound, and how things will take place, how it unwinds and how things take place. Okay, now let's try to know simple basics about the DNA replication now. Okay, now I want all of you to pay attention here. The two daughter strands, daughter means, the new strands. This is why this is one daughter strand. This is like a parent strand. This is a daughter strand. This is a parent strand. This is a daughter strand. Okay. So the two daughter strands are synthesized in totally different ways on the DNA. What does that mean? Now I want everyone to please pay attention. Let me erase this first. Okay. On one strand of the DNA, this strand, on one strand of the DNA, what happens? DNA synthesis is continuous. What do you mean by continuous? That, as you can see here, DNA is continuously formed without any fragments. Okay. And this strand, this new strand, is called as a leading strand. So the question says that what is a leading strand? You will say that the leading strand is that strand of the DNA which is continuously formed, which is continuously synthesized. Okay. And one thing I want all of you to remember this, that DNA synthesis always and always and always takes place in one direction, and that is 5' to 3' direction. I don't need to explain you what is the meaning of 5' and 3'. We already learned that in previous lectures. What is the meaning of 5' and the 3', right? Why do we use a dash over here, right? So I'm not going to explain that once again. We already discussed that. Okay. But remember this forever. The new strand of DNA is always synthesized in 5' to 3' direction. Are we okay? Okay. And I'll tell you why once we discuss the DNA polymerase enzyme, and I'll tell you why it is only 5 to 3' direction. Okay. Are we okay till now? Did you understand what is a leading strand? Leading strand is that strand of the DNA which is continuously synthesized without any breakup over here, as you can see over here. There's no breakup over here. And as you can see here, it's synthesized in 5' to 3' direction. But that is not the case with the other strand, because we know that the other strand needs to be synthesized again in 5' to 3' direction, right? So if you see here, other strand needs to be synthesized in 5 to 3. So what happens? The other strand is synthesized in smaller fragments. Okay, so this strand, which is not synthesized continuously, which is synthesized in smaller fragments, we call that strand as a lagging strand. And these fragments, these smaller DNA fragments, they are called as an Okazaki fragments, because they were named after the Chinese person who discovered these Okazaki fragments. Are we okay till now, guys? I'm going to ask you the most difficult question now. I know that you all will be confused now, okay? Give me a minute. So what did I say? I said that the one strand is synthesized continuously, okay? It is called as a leading strand, right? And the other strand is synthesized discontinuously. It is a lagging strand. So DNA synthesis always takes place in 5' to 3' direction. You have to remember this forever, because we'll discuss this when we'll discuss DNA polymerase. Because DNA polymerase can extend the DNA only in this direction, from 5 to 3', never from 3 to 5. Okay? But when it comes to reading, Doctor, the number of strands from where to where? If you see here in the leading strand, okay, if you see here, Faiza, in the leading strand, if you see here, okay, that's my next thing, which I'm going to explain. Okay, it's 5 to 3. Now, when it comes to reading the parent strand, okay, the parent strand is always read, read by the DNA polymerase in the 3' to 5' direction. Did I confuse you now? The parent strand, which is read by the, because see, DNA polymerase is the enzyme which will form the new DNA strand. But before this polymerase can form the DNA strand, it has to be attached somewhere, right? So for, let's say, DNA polymerase enzyme was attached here, right? So DNA polymerase enzyme can be attached only to 3' 5' direction, because it can read only 3' 5' direction. But when it comes to forming the new strand, that will always take place from 5 to 3. Same thing with the other strand. So DNA polymerase is going to read from 3 to 5, reading from 3 to 5, okay? So it's going to be attached over here, but when it comes to forming the new strand, it will form from 5 to 3. And I know this is something which everybody gets confused with, but I have a shortcut for this to remember, okay? Just remember this for your exams, and this is definitely a wonderful shortcut, which can, if it goes into your mind, you can never forget that. If there's any question about, how the DNA strands are being read by the DNA polymerase. So remember that, okay, how to remember, what is the, shortcut to remember this, okay? What is the mnemonic to remember this, okay? We always, if you have to prepare for the exam, what do you do first? Do you write first or do you read first? What do you do, guys? if you're preparing for the PHSF exam, which is coming next week, what would you do first? You will read the material first, right? And then you might end up writing down if you wish to write down, right? Remember that. So when you read something, okay, when the DNA polymerase reads something, you read first, right? And then you write down. When you read something, remember reading is always done from 3 to 5 by DNA polymerase. 3 to 5 directions. So reading done from 3 to 5. Reading done from 3 to 5. But when it comes to forming the new strand, so you write after the read, you know, you write something after you read something, right? So when you have to write something, what we're going to do, you write after you read, right? So when you write something, you write always in 5 to 3, in 5 dash direction 2. I'm sorry about my pen. I'm not able to write clearly with this pen. Are we okay about it now? You will, if you will remember this read and write formula, you will never ever forget. If I ask you to identify which strand is the, with respect to which strand the new DNA molecule is produced. Did it make things easy for you now? Did you all understand now what is this read and write? So the parent strand is always read from 3 to 5. Okay. So in parent strand, there's no question of leading and lagging. But when it comes to forming a new strand, then you remember it's always 5 to 3. This is always 5 to 3. So that is why I have put here, DNA is read by DNA polymerase in 3 dash to 5 dash direction. Okay, that means the new strand is produced only in 5 dash to 3 dash direction, because the new strand has to be anti-parallel. If this is from 3 to 5, obviously the other one should be definitely from 5 to 3, right? Because two strands are anti-parallel. Same thing here, 3 to 5, and the other strand is 5 to 3, right? And the second thing which I told you, that remember that on one strand, the DNA synthesis takes place continuously. That is a leading strand, okay? And on the other strand, the DNA synthesis takes place, you know, in small fragments. That is called as the lagging strand. Mr. Kazi, is it clear for you now? I think you asked a question earlier. Is it clear for you what is a leading and a lagging strand? And for everybody as well? Thank you, Mr. Kamya. Unfortunately, we could not see the videos, but I'm sure that, you know, once you see the videos, things are pretty much clear, you know, you see all these things happening animated, okay? Okay, that was about the basics of the DNA replication. Now we'll talk about the replisome. What is a replisome or a replisome? Replisome is that, you know, DNA synthesis is not an easy job to be done, okay? We need so many enzymes, we need so many proteins, okay, so that the DNA synthesis can take place. So these are the enzymes. So you have to memorize this table as well, okay? So these are the enzymes which we use in the DNA synthesis, like your helicases, primases, like clamp protein, DNA polymerase, and ligases. We'll go now one by one to each and every enzyme and how exactly the DNA replication takes place, okay? Step 1 of the DNA replication. If you remember, we spoke about, you know, how your, remember how your histone proteins, okay? If you remember this diagram, you know, when we discussed, do you remember this one? This one, how histone proteins, right? And how your DNA, which, you know, which is wrapped around histone proteins, right? If you remember that day, I told you that histone proteins are positively in charge. I'm not going to repeat why they're positively. We discussed previously about the side chain of the amino acids over there. And we know that the DNA, because of the phosphate, it is negatively charged, right? So that keeps your nucleosome, you know, that's how your histones and your DNA hug each other, because one is positive charge and the other is negative charge. So now when you have to replicate the DNA, okay, you don't want nucleosome. You want to open up the nucleosome, okay? You want a stage which looks like something like this, okay? You don't want this. You want DNA to unwind itself so that we can have a DNA like this, okay? There's no nucleosome, and then only the DNA replication will take place. Do you all agree? So don't you think so? This is the first step to remove the nucleosomes, right? To have this helical structure, right? So that we can start the replication, right? So how will that happen? That's going to happen by the opening up the superstructure of the chromosome. This was a superstructure, isn't it? We discussed about how loops and all these form the superstructure of DNA, right? So, so first step will be, we will do acetylation, okay? So what is acetylation? So in the first step, in order to remove the superstructure of the DNA, of the chromosome, to remove the nucleosomes, to open up, to open up histones and the DNA, okay, to remove the super condensed structure, okay, what do? We do something called an acetylation, okay. The histone, the lysine residue, in the histone proteins, you have a lysine residue, right? So the lysine residue of the histone proteins, okay, is acetylated, okay? And then we have an enzyme here, which is called as an histone acetylase, okay? Histone acetylase, we call it sometimes HAT enzyme. Histone acetylase transferase is an enzyme, okay, which causes acetylation of the lysine residue of the histone. What does that mean? That means acetylation, addition of CH3COO to lysine, okay? Lysine, which is basically present in the histone proteins, right? It removes some of the positive charge of the histone, okay? So as you remove the positive charge of the histone and DNA are going to separate from each other, because now the positive and negative, because unlike charges attract each other, right? So when you remove the positive charge from the histones, right, it will decrease the interaction of the histone with the negatively charged DNA. Did you all understand this? Why do we do acetylation? And we are not doing this. This is basically done by the cell, so that the supercoiled structure of the chromosome can be, uncoiled, and we can have a simple double helix structure of the DNA. How many of you did not understand this? Is it clear to everyone? So what will we have after acetylation? We'll have a more relaxed structure of DNA with no nucleosomes. There'll be no nucleosomes now, right? Okay, that was step one. Step two, I had a beautiful video for this. I'll just try to play this, because when you play a video, it becomes more clear. I had a video showing this coiled structure of DNA. Give me a minute. Let me give one more try to this, and let me see if I can play that. Hopefully I should be able to. Let me see. I'm going to give it a last try. Okay. Topoisomerase.
Let's consider what happens as DNA
Okay. Hopefully it should work. Share the screen. Oh my God, I cannot find the video anywhere. Okay. Tell me if you can hear the sound, and
Let's consider what happens as DNA unwinds during replication.
guys, can you see the video as well? What's wrong with it today? It's okay. Hopefully it will work now. Can you all see the video, guys?
As DNA unwinds, it acts like the... Let's consider what happens as DNA unwinds during replication. As DNA unwinds, it acts like this rope when we pull apart its two strands. As you pull the strands apart, twisting tension builds up in the rest of the coiled portion. It is actually adding one twist to the remaining rope for each twist pulled out of it. At some point, you can't separate the strands anymore. The remaining rope is too tightly twisted. If you relax your tension on the rope, it will twist around itself in a supercoil. It is releasing tension. If you want to keep pulling the rope apart, you have to release the tension periodically, and one way to do this is to cut the rope and splice it back together. This problem has been best characterized in small circular DNAs. There are two methods of dealing with this problem in DNA. One cuts only one strand of the DNA double helix, and the other cuts both strands. Let's look at the first. Topoisomerase I enzymes cut a single strand of the double helix, pass the other strand through the cut, and reseal the break, relaxing the overwound molecule, which now has one fewer twist. Topoisomerase II enzymes do the same thing but with both strands of the double helix. Topoisomerase II cuts both strands of a double-stranded DNA and passes another double strand through the break and then reseals the break. So if a molecule of DNA is supercoiled, topoisomerase II can remove the supercoiling, 2 twists at a time, to yield a relaxed circle.
Guys, were you able to see the video now? Alhamdulillah, finally we won. Alhamdulillah. Okay, okay. So what did you see? What is the job of the topoisomerase? Can you relate it now? What is the function of cut the DNA, unwind? Unwinding is different. No, unwinding is a job done by an enzyme called as a helicase. Enzymes are equally very important to convert the supercoil to a relaxed form. Absolutely right. And that happens when the topoisomerase is able to cut the DNA, make either a single strand cut or a double strand cut, right? So we have two types of we have two types. Relax the DNA, absolutely. Release the tension by cutting the strand, either one strand or both the strands, absolutely. First thing, topoisomerase is also called as a gyrase. And we have two types of topoisomerase. Topoisomerase 1 and topoisomerase 2. Topoisomerase 1 causes one cut, okay, and in a single strand DNA, while as topoisomerase 2 can cut both the strands of DNA, right? And what is the function of the topoisomerase? The function of topoisomerase is as the helicase enzyme, which we'll talk about in a while, as it tries to unwind the double helix DNA, right, as the two strands are separated. So there, as you saw in the video, it will result in the formation of positive supercoils, right? As the replication fork is moving forward, right, helicase is binding these hydrogen bonds over here, right? So there is a chance that the DNA can be supercoiled, right? We call it a positive supercoiling, right? So in order to remove this positive supercoiling, the topoisomerase moves ahead of the DNA strand, okay? It keeps the strand ready, okay, so that the replication can smoothly take place. So it can cause either a single strand break or a double strand break, and that's how it removes the supercoiling of the DNA, and the DNA becomes more relaxed after this, right? But remember that it not only cuts the DNA, it cuts the DNA as well as joins it back again, right? So this is done by the topoisomerase. Every enzyme of the DNA replication is important, right? In terms of, any question might be asked about it. Are we okay about the topoisomerase? Shall I move to the next enzyme? Okay, helicases. Okay, in order to understand the helicases, we need to go back to the structure of the DNA. If you remember, we said that two strands of DNA are holded to each other, something by the hydrogen bonds. Remember, the two hydrogen bonds between adenine and thymine, and three hydrogen bonds between guanine and cytosine that holds the two strands of DNA together, right? We spoke about that previously, right? So knowing that there are hydrogen bonds between the two strands of DNA, so what does a helicase do? Helicase is called as a DNA unwinding enzyme. Okay, it untwists the DNA. Absolutely, Mohammed. It untwists the DNA. It unwinds the DNA, right, by simply breaking these hydrogen bonds, right? And it's because of helicases, right, this replication bubble is formed when the replication is taking place. Or this fork, there are two replication forks over here. Two forks are formed. It's only because the helicases have attached themselves to one strand of the DNA. Okay, if you see here, there's a diagram over here. This is a diagram from your book. Look at this. Okay, where are helicases? Look at this. One helicase is bound to one strand, another helicase is bound to other strand, right? As they're attached to the DNA strand, they cause separation of the DNA strand by breaking the hydrogen bonds. You can think helicase like a zipper of your jeans. If you're wearing the jeans, okay, while wearing, you remove the zip, you pull down the zip, right? The way you pull down the zip and the zip opens, right? And similarly, it is an unzipper. Okay, helicase is an unzipper which unzips the two strands of the DNA. Are we okay about the helicases? Any questions about it? And then we also have a clamp protein. Okay, if you see the diagram over here, clamp protein is this one. Look at this. This is a clamp protein. Okay, this is a clamp protein. Clamp protein is something like it attaches, or it braces to the DNA strand, okay, and holds this linear DNA strand, which is no more now a helical strand. It's now a linear strand. Okay, it holds this DNA strand so that the nucleotides can be added. Nucleotides can be added to the new strand of the DNA. Okay, so that is the function of the clamping protein. Okay, to hold the linear strand, the straight strand of the DNA. That is the clamping protein, because the clamping protein is right over there in your this table. So you need to know this table. Then comes your another enzyme called as an RNA primase. Okay, before I could explain you about the RNA primase, what you need to know is that you cannot start DNA synthesis unless and until you have a short primer. What is a primer? Primer is basically a short sequence of DNA from 5' to 3' OH end. Okay, from 5' phosphate to 3' OH end, right? A very short sequence of DNA, like 4 to 15 nucleotides long, and DNA polymerase can add the nucleotides to this 3' OH end. The problem with the DNA polymerase is that it cannot start the synthesis of a new strand unless and until you have a short primer over there. It's something like, if you are in a ground floor, okay, you're from outside in your garden, and from your, you want to climb from ground floor to the second floor, okay, you will not be able to climb, right? What will you keep? You'll keep a small ladder, okay? So as you keep the ladder, and you can keep on climbing on that ladder and go to the second floor, right? Same thing, the DNA primer is basically a small ladder of the nucleotides, okay? and RNA polymerase adds nucleotides to this 3-OH group like this. That's how the new strand will be made longer and longer and longer. And remember, this RNA primer is never a DNA strand. RNA primer is always made up of RNA. So we will not have a sugar called as a deoxyribose here. We will have a ribose sugar here. Are you okay about the primer? And it's called an RNA oligonucleotide. What is oligonucleotide? Oligo means a short sequence of the RNA nucleotides. As you can see here, look at this. The green one is the primer, and then you have a leading strand. The green one is a primer, and then you will have this lagging strand, right? Then the DNA polymerase can extend the DNA. I think we'll stop over here. We didn't even finish the chapter today. Yeah, if we have the incorrect replacement, Mohammed, if there are incorrect nucleotides, it is a job of DNA polymerase, right? We'll talk about that as well, how your DNA polymerase remove those incorrect bases and replaces it with the correct bases, right? So proofreading, you know, for example, when you write your question paper, right? What do? At the end, you check your paper if there's any mistake, or you might have ended up, you know, instead of A, you wrote B, right? Same thing is done by the DNA polymerase. Once the replication finishes, it will definitely do the proofreading before it can proceed further. Okay, Mohammed? Oh my God, we couldn't finish the chapter today. But anyways, we'll just take, within half an hour on Wednesday, we'll finish the chapter and we can start with the transcription chapter. Okay. So did you all understand how was the class today? Was it boring? Was it interesting? Or, you know, how did you find the class? Was it interesting? I know this chapter is a little complicated, but did the information stick to you? Very interesting. Oh my God, really? Mashallah, very good. Wonderful. You know, I used to teach human genetics.